The significance of this protocol is that it allows an accurate rheological characterization of non-Newtonian biological fluids such as mucus, especially when only very small sample volumes are available.
This technique facilitates the characterization of the apparent yield stress and viscoelasticity of complex structured biological fluids, such as Gill-Raker mucus.
This method enables the yield stress estimation of Gill-Raker mucus and facilitates the rheological characterization and protocol development of similar biological fluids, such as human, animal, and plant secretions.
Demonstrating the procedure with Kartik Bulusu will be Samantha Racan, a graduate student from our laboratory.
To prepare approximately 2 milliliters of 100 milligrams per milliliter concentrated fish mucus solution, perform serial dilution of 2 milliliters of 400 milligrams per milliliter concentration by adding deionized water in 1:2 ratio twice. Place the vial of mucus solution onto a shaker until the solution is adequately homogenized and any mucus particulate agglomeration has been mitigated. To calibrate the rheometer, launch the rheometer instrument control software and select the Calibration and Instrument tabs. Click Calibration and Calibrate to ensure that the calibration values are within 10% of each other, and click Accept. Turn the shaft on top of the rheometer to install the cone angle geometry. The software will detect the 40 mm diameter, 1 degree, 0 minute, 11 second cone angle geometry.